The following program is part of a college course. These telecourses allow you to earn college credits at home. For more information, contact a college or university near you.
Life, in all its forms and functions, depends on a common process, cell division. With this remarkable force, nature delivers the genetic messages which define each organism, how it grows, repairs itself, and most importantly, how it produces the next generation. This has been true since the cycle of life began.
One of the things that must have happened very early in the evolution of life was a mechanism that allowed this primitive little glob of protoplasm, whatever it was, to produce two primitive little globs of protoplasm, and they in turn would produce two, each would produce two more, and so on. Once that happened, then life was on its way. So you really did have to have cell division as, or something like it, as one of the prerequisites for the beginnings of life, for the beginnings of the whole evolutionary story.
Cell division sustains all life, ensuring a continuous stream of genetic information for generations to come. The Great Exposition of 1851 produced a grand celebration. Outside London, the nations of the world gathered to display their wares and praise their industrial might. Still, the hardware unveiled in the Crystal Palace would not affect humankind nearly as much as the biological knowledge gained during the years that followed. Pasteur discovered germs. Lister taught us how to kill them. And Muybridge revealed the synchronized beauty of muscle and movement. Yet biology's brightest moment came when chemistry brought us color. In 1876, a German anatomist named Walter Flemming began using new synthetic dyes to stain tissue samples. Staining increased contrast, allowing him to study more clearly the miracle of cell division. For the first time, Flemming observed the central characters in this life-giving process, the nuclear bodies we now call chromosomes.
What Flemming saw, and others, was that chromosomes were sort of globular structures that changed their image throughout different portions of the cell cycle.
Flemming called the stained nuclear material he saw chromatin, from the Greek word for color. While he didn't name the larger objects he observed, those who did followed Flemming's lead and called them chromosomes, meaning colored bodies. Though he left many questions unanswered, Flemming offered our first glimpse at how cells pass their genetic instructions from one generation to the next.
When Flemming was doing his analyses of cells and chromosomes, he was limited to having to draw what he saw on paper and transmit the information and collect the data by that crude method. Today, we capture our images in a very different way.
With modern microscopy, scientists now view these genetic messengers in much finer detail. In each chromosome, we see two identical DNA molecules coiled tightly into strands called sister chromatids. Forming their familiar X shape, the chromatids connect at a single point called the centromere.
Centromeres are present at particular locations in chromosomes and are responsible for the movement of chromosomes. Without centromeres, normal chromosome inheritance would not occur, and abnormalities would occur that would kill a cell. A subject of investigation at this time is to try to understand what the in-depth structure of the centromere is. What are the DNA sequences that are responsible?
Researchers have just begun to unravel the mysteries of chromosomes. The most astounding of these includes how our complex DNA molecules can fit into such tiny bodies.
If you summed up the entire length of all the DNA molecules in a cell, you'd be able to stretch them out and they'd find they were about a yard long, and yet a cell is below the level of visibility. How does a chromosome do this? Well, the DNA, as you know, is made up of a double helical molecule. The double helix itself is wrapped into larger helical molecules, which in turn are wrapped again into more complex structures. And all this is done as a result of the interaction of DNA with certain proteins that bind very specifically to the DNA and give the entire chromosome its structure.
When researchers examine all the chromosomes from one cell, a process known as karyotyping, they reveal a variety of different sizes, shapes, and patterns. Yet even in this genetic puzzle, we find matching partners.
All humans have 46 chromosomes, and those 46 chromosomes are actually two sets of 23 chromosomes each, which are inherited, one set from your mother and one set from your father. Now, when we stain these chromosomes, each chromosome pair can be matched up because they have identical banding patterns. And these pairs of chromosomes that have the same shape, size, and bands are called homologous chromosomes.
In addition to identifying these homologs, karyotyping reveals the number of chromosomes. Members of the same species, such as this flock of flamingos, all carry the same number of chromosomes in each cell. But what of different species?
The number of chromosomes varies from species to species and does not directly correlate with the complexity of the species. For instance, in yeast, which is a small single-celled organism, they're in the neighborhood of 13 chromosomes, while in Drosophila, a multicellular organism, a fruit fly, there are only 4 chromosomes. However, the information content in each of these chromosomes is significantly different. The chromosomes in the yeast are very small. They're more numerous, but they're smaller, whereas in Drosophila, they're much longer and much larger, and they contain many more genes.
So, a high number of chromosomes does not necessarily produce more complex organisms. While humans have a total of 46 chromosomes in each cell, a toad holds just 22. Yet a simple plant, the horsetail, carries 216 chromosomes per cell. Regardless of their number, these chromosomes hold the life-giving secrets locked in our DNA, secrets which must be passed on to future generations. This occurs through 2 different methods of cell division called mitosis and meiosis. From the dawn of their lives, all complex organisms rely on cell division to grow and heal. But to maintain a consistency of traits, each cell must pass on exact copies of its genetic instructions. In the wine business, this consistency can mean the difference between success or failure. Winemakers spend years developing specific grape varieties, cultivating the special characteristics that make a fine wine. Once selected, whether Cabernet or Merlot, they work to keep each year's crop the same as the last.
Winemaking is mostly chemistry, and winemakers are mostly chemists, and they're playing with flavors all the time. if one lot of Merlot didn't taste that well, in their opinion, they'll add 2 or 3 blends of different clones of Merlot, and it may be that great bottle of wine.
To maintain this quality year after year, winemakers reproduce the best vines with each new season. Some varieties trace their lineage back nearly 150 years to European vineyards. Each vine is a clone, the product of an uninterrupted sequence of identical cell divisions.
Clones come from a single source, and those sources then go to nurseries, and then from there you know that when you're calling up for a certain clone number that your neighbor has and you like that style of wine, you can be pretty set that it's going to give you pretty straightforward kind of grapes, and kind of, well, it'd be for sure, because it won't change from your neighbor's to yours, as long as it's the same clone.
Winemakers maintain the consistency of their clones through the process of grafting.
Grafting essentially is taking a rootstock that is viable and grafting on a clone. You could take a Chenin Blanc rootstock or a Sauvignon Blanc rootstock, and you could graft on top of that with scion wood of Merlot, and it would take. The scion sticks will have the same identical gene code. At Callaway Vineyard Winery, when a wine change needs to be made, we first make a grape change in the vineyard. Here you can see I've made an upcut exposing the cambium layer of the rootstock here. I will then take also a scion wood of the clone that we want to change to. And when I do that, I put the 2 cambium layers together of the rootstock and the scion wood, and there's where the mitosis operation takes place.
The success of this graft depends on the cells of the root and vine dividing and growing together. During this process, each cell will pass through a chain of events called the cell cycle. This includes 2 primary phases, a period of growth followed by a period of division.
In a complex sequence of events, one cell brings life to 2 new cells. But before a new cell divides, it must make a few preparations during a part of its cycle we call interphase. First, the cell grows, building up the molecules and organelles it will need for the coming event. Next, the cell replicates the DNA in its nucleus. This assures a complete set of genetic instructions for each new cell to come. Finally, the cell forms the barrel-shaped centrioles and protein fibers, which will serve a key role in the division process. With all in place, interphase is complete, and the cell is ready for the next stage in its cycle of life, mitosis.
Well, interphase looks like a very boring stage in the cell cycle. You don't even see chromosomes. Doesn't really look like a lot is happening, especially in comparison to mitosis, where things are being torn apart and moved around. But in fact, in interphase, as we now know, there is an awful lot happening. In fact, that's when much of the business of the cell is occurring.
A cell in your body that's dividing rapidly, or is in a tissue that is dividing rapidly, like, say, a cell in your bone marrow or something like that, takes about 24 hours to go through a complete cycle. And that complete cycle includes the growth of the cell itself, the duplication of the DNA in the cell, because each DNA molecule must make a copy of itself at some point during the cell cycle, and then finally the process of mitosis that distributes the chromosomes of the cell to each of the daughter cells. And all these things take up the full 24-hour cycle, of which only about 1 hour is actually involved in the cell division process.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
Underneath this tape is where we will see mitosis in action here. And notice all this puffiness here. That is the callus cells. Callus cells will only happen when the two cambium layers are intact with each other. If you can imagine, if you cut your finger and the callousing forms and heals that wound, this also was a wound in terms of this vine. The vine now is happy again. It has sealed its wound, and this is mitosis at its best right here. Shows that we had a successful graft. Everything's going to be fine.
In all complex organisms, mitosis drives the growth of new tissue and the repair of damaged parts. But some depend on this process for much more. Many plants and even some animals reproduce asexually, without a partner. Using mitosis, they produce offspring which are exact copies of themselves. Genetic consistency offers some advantages in nature and makes a fine glass of wine. But for reproduction, many life forms encourage variety over consistency. These organisms require a special type of cell division to complete their cycle of life.
It's very good.
See, it's also watching us.
We expect zoos to display animals, but with many species facing extinction in the wild, some zoos have begun breeding programs as well. This presents an intriguing challenge.
The background knowledge to breed animals at will, to prevent that they die out from inbreeding or from diseases, is very difficult to obtain. Not enough knowledge is had, for instance, of the biology of giraffes, of elephants, or of polar bears to control breeding or to enhance it. That is not had for most zoo animals.
As zoo biologists struggle to repopulate exotic species, they often encounter unexpected problems. For example, when low reproduction rates plagued a species of West African antelope called the dik-dik, researchers uncovered a case of mistaken identity.
We first got involved with working with dik-diks through another zoo who had problems identifying what species they had. And we proceeded to take samples from those animals, and from these we were able to make chromosome preparations and analyze these under the microscope. When we started looking at them, we found a vast array of chromosome numbers and sizes and shapes, and we were just very perplexed.
Individuals from the same species should carry the same number and type of chromosomes, but these samples did not match.
One had 46 chromosomes, and the other had 46 and 47 chromosomes, depending on whether it was a male or female. So what we were calling a single species actually were different genetic populations.
With the discovery of chromosome variations, scientists confirmed not one, but 2 species in this group of antelope. To reproduce successfully, organisms must combine matching chromosomes during sexual reproduction. This requires the fusion of special cells, an egg from mother and sperm from father. Each animal produces these cells only in sex organs, using a special form of nuclear division called meiosis.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The 2 homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in 2. This second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
Meiosis is a very unique process, and it happens only in the reproductive cells, the gametes, the sperm and egg cells. And it happens because there needs to be a reduction in the amount of genetic material that is passed on as an egg or a sperm.
When sperm and egg unite during fertilization, they combine their chromosomes, producing the correct number for that organism. They also bring with them the variety of genetic traits that were shuffled during meiosis. This variation offers a substantial advantage in the struggle to survive.
Everybody has a different nose and a different hair, and that's how we recognize each other, because the information contained on our chromosomes is slightly different, and the mix is slightly different. And the beauty of this is, and the reason for it is, that it allows all of us to react slightly differently to environmental insults. If all of our genes were the same, a slight change in temperature, or the unavailability of a certain vitamin, or the acquisition of a new virus would kill all of us.
Individuals of the same species show amazing variety, yet their cells hold chromosomes of the same number, size, and shape. In the case of the dictycs, while they may look the same, they represent 2 different species with different sets of chromosomes. Their troubles began when the 2 species interbred to produce hybrid offspring.
We took testicular biopsy samples from hybrid animals, the hybrid Kirk's A and Kirk's B dictyc, and looked at that under the microscope and discovered that, in fact, because of the different chromosomal rearrangements between these 2 populations, there was meiotic disruption. And you can see within the tubules here, there's no meiotic activity. The center should be filled with sperm, and essentially it renders this animal infertile. He's sterile, just like a mule.
Meiosis was not occurring in male dictyc hybrids. The evidence was conclusive. There could be no reproduction. The case of the sterile dictyx illustrates the crucial role of matching chromosomes during sexual reproduction. And while problems can occur, the long-term advantages over asexual reproduction make the process worthwhile.
Asexual species simply don't last as long as sexual species do. Asexual species go extinct sooner than sexual species. The question is why? Well, one answer to this is that conditions do change. And if you're beautifully adapted to one set of conditions and all your offspring are identical to you, then if conditions change, this could be a problem, because if all of a sudden the conditions have changed to the point where your offspring are no longer well adapted, then you can see that, that they're going to be at a disadvantage compared with a sexually reproducing population that produces a great variety of different offspring.
Nature builds this great variety using complex molecular instructions stored in our DNA. But to continue this engineering marvel, these instructions must be transferred individually, one cell at a time. This occurs through the miracle of cell division. Through its intricate processes, so remarkably dependable, all complex organisms build and sustain themselves, handing off their genetic secrets, passing life from one generation to the next.